Hello and welcome to Biology of Cells and Tissues. So this course, I'm just going to do a couple little housekeeping things. Biggest thing about this course, I'm going to talk about the lecture material, talk about the lab material, and the class as a whole. This class is very slide heavy. What that means is that all of the material that you're going to be quizzed on, that you're going to be tested on, is in the slides that the professors are going to provide. At least that is the case if you are taking this course at Parker under Dr. S. Okay? If you have a different professor, obviously, speak to previous students, speak to other people, and figure out kind of how that specific professor tests. Dr. S's slides that I have here are awesome. They have all the information, especially all the information, the extra tidbits and stuff that I have added into them is really, really good and will give you all of the information that you need. So, lecture material. Lectures are important, going through the slides, and again, I'm going to go through a lot of these slides. As much as I can, I'm going to try not just straight read from the slides. But these slides are really, most of them are very good and they flow very well in the sense that, you can just read straight through them. It's going to be a lot of definitions. That's kind of what this course does. This course is going to set you up for a lot of your future courses. There's a bunch of overlap with biochemistry, with anatomy, and future courses when you get into like microbiology, future biochemistry, physiology, all that kind of stuff. You're going to notice a lot of overlap from this course. As a result, because there is so much overlap, there are a ton of just straight definitions from this course. So that's what we're going to spend a lot of our time on, is just pinpointing those definitions and those words and familiarizing yourself with the science speak, as we would call it. Additionally, lab side of this course. Under the lab side of this course, it's essentially going to be a bunch of pictures. Let's pull up one of these labs. For example, all of these PowerPoints. Most of them are going to include images and pictures. These kind of images and pictures that you see, this is exactly how this is going to be tested. Okay? This professor specifically likes to pull the exact same pictures that they use in the PowerPoints in order to test. Let me see which one. I believe it was this PowerPoint. Yeah, so like these pictures here, they will pull a lot of these out of here. Now, the way that it's going to be like an arrow, kind of like this, a big arrow pointing to something, and it's like, hey, what is this? That's kind of what a lot of the questions are going to be on the lab side of this test. Now, the important thing to note is when you look at these, make sure that you are also looking at all of the other structures around. So, for example, this one right here is pointing at a soma, a cell body of a nerve cell. That is what we call a soma. But at the same time, you can also see the nucleus on each one, and then the little blue structure deeper is the nucleolus. So you're going to have, multiple other structures on a lot of these that maybe aren't necessarily specifically labeled. So like here we have these different arrows. We have a molecular layer, we have a Purkinje layer, and the granular layer. So a lot of what the review is for the lab portion is looking at pictures and labeling everything and figuring out what is what. If you purchase my notes, which again the link is in the description down below, all of this is already labeled for you. So you can use this as you practice. Okay, so this kind of review, this kind of stuff is really, really great for practicing all of this material, understanding what is what. And this course, again, like I said, really sets you up to do really, really well in future courses because you get familiarized with a lot of the histology meaning the different cell types, as well as a bunch of the anatomy. Now, without further ado, let's jump into kind of week 1 material. There's a lot of it here. So, what I'm going to do is I'm going to go ahead and set a timer. We're at 5 minutes right now. We're going to go until 30 minutes, and then at the 30-minute mark, I will end this video and make a part 2. My guess is that this might take us 2, potentially 3 parts to get through all of this material. Okay. Talking about macromolecules. This is another little note. I want to make sure if you have purchased my notes, again, link is in the description to get to those. I like to include on the PowerPoints lots of text information down in the bottom here. If you are taking my notes, converting them to PDF format, and then uploading them like into GoodNotes or Notability or whatever those are for the iPad, I know a lot of people prefer to take notes that way. If you are doing it that way, make sure that when you convert everything, so you can go file print or even to export, when you go to do this, make sure that you include it more like a notes page, so that way anything that are notes underneath are going to also be included. That is the easiest way to do that. That way you still have this whole thing. You can zoom in, pinch, zoom in, and write on the slide, but you get all of my additional material down below. Okay. All of our bases are covered. Let's jump into macromolecules. Again, we talk about macromolecules. These are our large, complex molecules. We're mostly looking at like the proteins, lipids, carbohydrates. Kind of all of those will eventually come together in different ways to build up and create bigger molecules, bigger structures, bigger things in the body. And we're looking at essentially the overall structure of the cell in this lecture. 3 objectives: understand cellular structures in molecular terms, understand the chemical nature of monomers involved in biosynthesis of macromolecules. We'll see monomers again in just a minute. Monomers are like the very basic singular units that we're going to be putting together. Think of it like a puzzle. A monomer is a single puzzle piece, and the macromolecule is the completed puzzle once all the monomers are put into place. Describe the synthesis and properties of macromolecules of the cell. We'll look at like kind of what their functions are, what their jobs are, why they are important. So, the macromolecules are responsible for most of the form and order of living systems, are generated by the polymerization of small organic molecules. Polymerization, poly, meaning many. The merization portion is kind of combination or fusion. This might date myself a little bit. When I was growing up, I watched a lot of Yu-Gi-Oh! In Yu-Gi-Oh! There is a card called Polymerization, which fuses other stuff together. If you ever watch that show or you're a little bit of a nerd like myself, maybe you get that reference and that helps you remember that. But we take a bunch of monomers, so little individual things, like one of these little guys, and we put them all together to make bigger things. That is polymerization. The repeating units are going to be called monomers. That's one single unit. So, such as glucose being present when you talk about sugars or starches. Amino acids come together. An amino acid is the monomer, and that all forms together. It is polymerized to form proteins, and nucleotides are all strung together to form nucleic acids. And we'll look at each of those individually throughout this PowerPoint. The importance of synthesis by polymerization. So synthesis, so creation by this joining together of all the monomers. Most cellular structures are made of ordered arrays of linear polymers called macromolecules. So macromolecules are polymers. I'm going to highlight that polymer as well. I'm going to, if you downloaded my notes previously and you don't see some of these edits, once I finish this recording, I'm going to save this and go back and upload the updated version of it. Like I said, as I continue to make these better, you will have continued access to them. So if you see me coming out with new videos and stuff over material, more than likely you will also again see updates and stuff to the material itself. So just pay attention to that. Okay. Important macromolecules in the cell include proteins, nucleic acids, and polysaccharides. We'll get into all of what those are in a little bit. Actually, let me do this just to keep this so you guys get a little peek into my mind. I'm going to put everything in here, and we'll keep adding more and more to this. So we look at what a protein is. The protein is a molecule made up of amino acids. It's needed for a lot of functions. We're going to spend a lot of time nucleic acid. We're going to spend a ton of time talking about proteins. Proteins make up a bunch of stuff in the body. Very, very important. Nothing really happens without proteins. So a good chunk of this course, as well as biochemistry, a huge chunk is literally just talking about different types of proteins and what those proteins do and how they function in the body. Then I threw another definition of nucleic acids as well as polysaccharides. Nucleic acids are large biomolecules. They play essential roles in all cells as well as viruses. This is nucleic acids. We're talking about our DNA, RNA, what makes us us. And then polysaccharides, long-chain carbohydrates put together of a bunch of monosaccharides. Monosaccharide is a monomer. It's just a sugar monomer. Typically used by the body for energy or to help with cellular structure. And we'll look at both of those. Here we are connecting some of the dots. So proteins is the polymer. Remember, polymer is just a string of monomers. So basically, if we have an amino acid, a single amino acid is this little circle. That is an amino acid. We string together a bunch of these. In real world, we're stringing together hundreds, if not thousands of these. Then the whole thing eventually becomes a protein. It's a little bit more complicated than that, and we will walk through the whole process of that in future parts of the material. Polysaccharides, polymers of sugars or carbohydrates. We'll look at the specifics of that later. Nucleic acids, polymers of nucleotides. Lipids are made up of a variety of different molecules, and we'll look at those. A lot of lipids. Lipids are fats. Lipids are our fats. Lipids play a very, very important role in lots of parts of the body. They're made up of different things, combination of carbons, sometimes of changed sugars, and amino acids that kind of mix together in order to create specific lipids. And the little detail, except for lipids, these macromolecules are also called polymers. So proteins, polysaccharides, nucleic acids. These are polymers. Lipids are just lipids. It's just another macromolecule. So we look at these carbohydrates, proteins, lipids. You should have, hopefully in undergraduate, seen some of this kind of stuff. These are our main, macromolecules that we consume. We consume sugars, we consume proteins, and we consume fats. This is what keeps our body alive. This is everything that we really, really need in our bodies, okay? Metabolism, when we talk about metabolism, you'll see this a lot in biochemistry. We're talking about like catabolism or anabolism. Anabolism is building stuff up, creating stuff, synthesizing. Catabolism is breaking stuff down into its individual components. So for example, like carbohydrates, if we actually look at all of these, the carbohydrates, the proteins, the lipids, if we were to break them down into their individual like atoms, we're going to see hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus. Like this is essentially what most of all of these are made up of. I'm not going to say this is absolutely all of them because there technically are a couple of other ones that we will see, but like these are all of our core components that make up all of these structures. Same with nucleic acids, the DNA, RNA, all of these. It's all of this stuff that's built up, or all of our DNA, is just a bunch of these all strung together. When we talk about synthesis of macromolecules, let me just make sure. Okay, we're going to get deeper into the proteins next. Okay. This is the general step-by-step overview of how this happens. How do we go from a single unit to a big long chain that does what it needs to do? So we take a monomer and we have what's called an activated monomer using ATP. ATP, you will hear tons and tons and tons about ATP. If you do not recall this, ATP, insert text box, there we go. ATP equals adenosine triphosphate. This is our energy source of the body. Pretty much all of our energy as well as any other kind of chemical reactions are coming, the energy that we need for it is coming from ATP, adenosine triphosphate. A lot of biochemistry this trimester, a lot of your units is going to literally just talk about how we make ATP for all of the other stuff that we need. Anyways, ATP is what activates the monomers. So monomers have available hydrogen, OH. OH is known as an alcohol or a hydroxyl group. So hydrogen, hydroxyl, again, it's more biochemistry type terms. These two are able to bind together easier than a lot of other things. So that's typically where we see stuff bind. So the monomer activation, so monomers, M1 and M2, with those available, are activated by coupling to the appropriate carrier molecule, just shown in purple, using energy from ATP or a similar high energy compound. So we have some kind of carrier protein binds with using ATP, and then those come and they bind together. Monomer condensation. Condensation is a type, this is more biochemistry, you'll see this in biochemistry. Condensation is a type of reaction. So it's a type of reaction where a bond forms and water is released. That's condensation. Again, in my biochem videos, I go deeper into all of those different types of reactions. Condensation is a very important one, where in the process of bond formation. So in this page, we'll want to remember that word right there, monomer condensation, the type of reaction that helps form bonds between monomers. And finally, this process just repeats over and over and over and over again until it elongates to the correct size. So here's just looking at some of the other steps, some of the key little details. Condensation, aka, put an aka here instead, aka dehydration. It's two words for the same thing. A condensation reaction equals a dehydration reaction, same thing. So the removal of water to allow for the bond must have reactive hydrogen and reactive OH on the molecule. So these are the conditions that have to be met in order for this bond to form. You have to have an OH, a hydroxyl group, and a hydrogen in order for that to work. They must be activated, so they have ATP, requires energy, ATP, and the components attached to carrier molecules directionally. So they build in a specific direction. They'll build, for in this case, left to right. Usually the way that we describe this, we'll have some kind of blank terminus to blank terminus. Like we'll say N terminus to C terminus, meaning that all the way on the left side at the very beginning, there is a nitrogen, and all the way on the other side at the very, very end, there is a carbon. So N terminus, C terminus, that's typically the way that we will see it described, depending on what this is going on. So in this case, like this would be directionally H terminus to OH terminus. Okay. A different kind of carrier molecule is used for each kind of polymer. You'll talk about this a lot in biochem, but just think of, so proteins and enzymes are often specific for specific jobs or functions, which means that we create proteins and we create enzymes, we create these things in order to do specific jobs, and they do those specific jobs. So we need different types of them, different kinds of them, to do all the different jobs in the body that's necessary to do. For example, for protein synthesis, amino acids are linked to carriers called transfer RNA, tRNA. We'll dive into that deeper in the future. Sugars like glucose that form polysaccharides are activated by linking them to ADP or UDP. So these are the specific carriers for the sugars. Nucleic acids. The nucleotides themselves are high energy molecules. Nucleic acids often have a phosphate group on them. So these are just examples: proteins, tRNA, sugars, ADP or UDP, nucleic acids, ATP or GTP. This slide seems like a lot right now because there's a lot of terminology that I'm sure you haven't seen before. Bear with me. You're going to see a lot of this in the future, okay? A lot of this right now is you need to get yourself familiarized with these terms and know what these terms are in general to be able to apply them in the future, okay? That's our main goal for this first chunk. Here's just, again, kind of a repeat of which molecules are important. We have the proteins, nucleic acids, and those polysaccharides. Okay, I think I'm going to actually stop the first recording a little bit early before the 30-minute mark, and we're going to start the second chunk with proteins. That way we keep it all together. So I will see you in the second part for the macromolecules.